Now we are going to learn about the small intestinal movements. Basically, there are two types of movements. That is, one, the mixing movements, it is otherwise called as churning. The other is peristalsis. So we'll see one by one. First, about mixing movements. So these mixing movements are carried out by the segmentation contractions. So these contractions occur as a result of the constriction of the circular muscle that is lining the gut wall. And what is the purpose of these contractions? So these contractions are non-propulsive. That means these contractions will not allow the food to propel forward into the next segment of the gastrointestinal tract. In turn, they will keep the food in the particular segment of the small intestine. By means of these segmentation contractions, the food is completely mixed with the digestive juice. So the main purpose of these mixing movements are that these contractions will bring about the mixing of the luminal contents with the digestive juice, so that the entire luminal contents are exposed to the digestive juices, and this will ensure complete digestion of all the food particles present in the luminal contents. So that is the main purpose of these segmentation contractions. Let's see how it happens. So first, there will be a set of contractions that will occur at particular points. So let's consider this is point number 1, this is 2, this is 3, and this is 4. At these 4 points, a segmentation contraction will occur, dividing the small intestine into small segments. So these are the small segments. Okay, so after some time, after few minutes, a new set of contractions will occur in the middle of each segment. So here, at points A, B, and C. So that this process will be repeating again and again until the luminal contents are dug in and rolled over and it is completely mixed with the digestive juices. So they will be repeating again and again. So new set of contractions will be taking place at different points until the luminal contents are completely mixed with the digestive juices. So there is also another purpose of this mixing movements. They will actually decrease the unstirred water layer. So this water layer will be present near the apical membrane of the enterocytes. So these mixing movements will actually decrease the unstirred water layer near the apical membrane of the enterocytes. Thereby, it will facilitate the absorption of the food nutrients into the enterocytes. So that's all about the mixing movements, and these mixing movements are caused by the constriction of the circular muscle present in the gut wall. So next we'll see about the peristalsis. So peristalsis, unlike mixing movements, peristalsis are propulsive movements. So this peristalsis will cause propulsion of the food from one segment of the intestine to the other segment in the oral to the caudal direction. Always the movement of the food bolus will be from the oral to the caudal direction, and that is called as law of gut. And what is the reason for this unidirectional movement of the food bolus is that the design, that is the gut wall, the myenteric flexes of neurons and the different set of neurons present in the enteric nervous system is so designed that the movement is always from the oral to the caudal direction, and that is called as law of gut. And now let's see how this peristalsis happens. See, the peristalsis is actually a reflex response that occurs when the gut wall is distended. So the primary stimulus for the peristalsis to take place is distension of the gut wall. So as the food bolus enters into the, as you can see in this picture, the food bolus is entering into the intestine. So because of the presence of the food bolus, the gut wall is distended. So you can see the distension of the gut wall. So this is the stimuli for the peristalsis to take place. Now we'll see what happens. See, as this local stretch, it will cause release of serotonin. So there will be release of serotonin at this place, and this serotonin will stimulate the myenteric group of neurons. So this myenteric flexus is actually present between the circular muscle layer and the longitudinal muscle layers of the gut wall. So the myenteric neurons will be stimulated. So these myenteric neurons contain cholinergic neurons. So the cholinergic neurons that are present behind the bolus will secrete a neurotransmitter called as substance P. and acetylcholine. So you'll be knowing about acetylcholine, right? So the neurons that are present behind the bolus will secrete substance P and acetylcholine. So this substance P and acetylcholine will bring about constriction of the circular muscle behind the bolus or in the downstream segment. Whereas the neurons that are present in the gut wall of the in front of the bolus will secrete neurotransmitters VIP and nitrous oxide. VIP is nothing but vasoactive intestinal polypeptide. So VIP is nothing but vasoactive intestinal polypeptide and NO is nothing but nitrous oxide. So these two neurotransmitters will bring about relaxation of the circular muscle in the upstream segment or in the segment that is ahead of the bolus. So as a result of this, there will be a constrictive ring behind the bolus and there will be relaxation in the upstream segment. And this will cause the food bolus to propel forward. So it will move forward and it is always towards the caudal direction. So these are the steps of the peristalsis. So the primary stimuli for peristalsis is the presence of food bolus in the intestine that will cause a local stretch and that will release serotonin. And this serotonin will stimulate the myenteric neurons and the cholinergic neurons present in the myenteric plexus. And these cholinergic neurons that are present behind the bolus will secrete substance P and acetylcholine, causing constriction of the circular muscle behind the bolus, and these cholinergic neurons that is present in the upstream segment will release vasoactive intestinal polypeptide and nitrous oxide, and that will cause relaxation of the circular muscle. And so these steps will propel the food forward, that is in the caudal direction, which is called as law of